Okay, vegetable-based fermented food. Normally, vegetables are healthier because of its nutrient content. Fresh vegetables. Normally, fresh vegetables are recommended. Normally, fresh vegetables are recommended because of nutritional value. Vegetable nutritional value like that, it contain good source of water, higher percentage of water, contain considerable amount of carbohydrate as well as small portion of protein. Out of 100% water in the vegetables contain 80 to 90%. So good source of water present and maximum portion is the water. Carbohydrate portion 2 to 6% and protein 1 to 2%. And rest of the percentage contain good source of fiber, vitamins, and minerals. So normally vegetables are very good, very healthier version for health. So why we need fermented vegetables? What is the beneficial effect of fermented vegetables? Try to answer.
শেলফ লাইফ বাড়ানোর জন্য।
আর একবার বলবে প্রিয়ম বলছো তো?
হ্যাঁ স্যার, শেলফ লাইফের জন্য একটা ইম্পর্টেন্ট ফ্যাক্টর পড়তে পারে।
Good point. First point is to increase the shelf life. Second point, Monomai, what you were saying about? Huh? Nutritional value. That nutritional value should enriched or increased by the help of fermentation. Any other point? Why we need fermented vegetables? Anyone?
স্যার ভেজিটেবলের গায়ে অনেক সময় স্পয়েলেজ কজিং মাইক্রোবস থাকে, তো ওটাও মরে যাবে।
After fermentation. So self life increase. That is same
হুম হুম।
point. Self life increase also same. So start the topic. Benefits of vegetable based fermented food. Fermented vegetables. Number one, increase the shelf life. And it is very simple. After the fermentation, various acids are produced. These acids actually uh, try to eliminate the other spoilage causing organism and the shelf life automatically increase on the vegetables. So first improve or benefit increase the shelf life. Second point, increase the consumption rate or ness, consumption ness means Normally, vegetables are season based, yes or no? Winter vegetables normally not available in the market, or the price of the vegetables will be rise in the summer season. Correct or no? But after in the fermented vegetables, the shelf life is increased, so we can use it for all over the year in the all season. That's why the concept is given. Vegetables, fermented vegetables are now grown, now produced, not grown. It is produced that we can use for all over the year, every season. Otherwise, the normally vegetables are normally season based, and after the one season, the vegetables will gone or it will be higher price in the market. So that's why the consumptionness is increased. That means all over the year we can consume particular vegetables by means of fermented vegetables product. So we can use fermented vegetables all over the season. Number three, higher nutrient value. Obviously, when the microbes, preferential microbes are grow, ferment the products, the nutrient value will be increased, more vitamins will be produced, more bioactive components are produced, and that help or benefited our health. So nutrient value increases. Number four, probiotics. The fermented causing bacteria usually use lactic acid bacteria. This lab and lab is a good source of probiotics, improve the gut health of the human body. That's why it is considered as probiotic bacteria. So fermented vegetables also good source of probiotics. Number five, improve taste. After fermentation, the organoleptic property changes or improved, and that increase the consumption is. That means taste will be changed, and many person can adopt that taste and can consume. And normally many people avoid vegetables because of taste. It's a bland taste. Many of times vegetables are bland taste. But after fermentation, the taste will be increased, and many person can use it, habituate it, and can use or taste it. So organoleptic property will be improved after the fermentation. So these are five beneficial effect of fermented vegetables. Shelf life increases, consumptionness increases, nutritional value increased, probiotics good source of probiotics, and taste also increased. In our syllabus, two fermented vegetable products are given. One is pickle, other is sauerkraut. And these two products are all over the world used. So widely used, fermented vegetable products are sauerkraut, pickles, kimchi. It is another popular Korean fermented vegetable food. And also in some country like USA, Western country uses olive, fermented olive. But in our syllabus, only given sauerkraut and pickles. In all type of fermented vegetable foods, fermented vegetables relies on natural lactic microflora. Natural lactic microflora to carry out the fermentation. That means similar here. Many of times do not use any starter culture. They relies on natural microflora. And which type of microflora? Lactic acid producing microflora. That means lab. That means lab lactic acid bacteria. And both homo and heterofermentative bacteria they are relies on homolactic and heterolactic.
তো এটা আরেকবার এক্সপ্লেন করবেন?
Yes, fermented vegetables production do not need any starter culture because microflora that is indigenously present on the vegetables are used to make the fermentation. And the fermented organism that we relies on lactic microflora. Lactic microflora means lab lactic acid bacteria. Both type of lactic microflora present, homolactic and heterolactic. That means it is considered under lab lactic acid bacteria. So here also we are relies on lactic acid bacteria to make fermentation to occur. Clear? But do not need any starter culture. This lab present indigenously, naturally present. So now take the list naturally occurring vegetables based microflora. Microflora on vegetables. If you listed down the microflora in vegetables, only labs are found or other non-lab microbes also found. So both lab means lactic acid bacteria and non-lactic acid bacteria. Both type of bacteria are found on the vegetables. Vegetables contain all type of bacteria, but we should select only lab and eliminate non-lab. And that is the condition during the fermentation it is achieved. By that condition, the lab will be selected and non-lab will be eliminated. So various kinds of microbes can be found inside the vegetables, but we should select lab. That is the conditional effect. So microflora in vegetables contain many of group of microbes like aerobic microbes, like enteric microbes, enteric bacteria, contain yeast and mold. Some examples: aerobic microbes present like Pseudomonas genus, Flavobacterium genus, Micrococcus genus, and Bacillus genus. Enteric bacteria like Enterococcus, Enterobacter, Klebsiella, and Escherichia. Yeast mold like Fusarium, Aspergillus, penicillin. They all are non-lactic acid bacteria. So this group A, B, C considered as non-lab, non-lactic acid bacteria, and also contain group D, that is lactic acid bacteria, lab, indigenously, naturally on the vegetables. And some of them are lactobacillus, streptococcus, pediococcus, leuconostoc. And group D is clearly lactic acid bacteria, lactic bacteria. That means produce lactic acid when they grow. So what is our priority? Which types of bacteria we have grown inside the vegetables based fermented product? Only lab, and try to eliminate the non-lab, yes or no? Otherwise, this non-lab create the spoilage. This non-lab actually the culprit of making different diseases, yes or no? So we had to eliminate this non-lab bacteria and select only lactic microbes. So how do you select lactic microbes over non-lab? Try to explain, try to answer. How do you select lab over non-lab? অনেক ফার্মেন্টেশনে পড়ে নিয়েছো, তো ট্রাই টু আনসার ল্যাবকে কিভাবে সিলেক্ট করবে নন ল্যাব থেকে। Because here we are do not using starter culture. Starter culture দিলে তো হয়ে যেত, আমরা ডাইরেক্ট দিয়ে দিতাম ল্যাব। We are not using the lab, sorry, starter culture. We relying on the natural bacterial process. So we had to select lactic microbes over non-lab and eliminate the non-lab. So কিভাবে হবে possible? How is it possible? Fermentation condition কিভাবে চেঞ্জ রাখবো? How do you modulate the fermentation condition that select lab only? টেম্পারেচার কে কি রাখবে, এলিভেট রাখবে কি না কমাবে? পাস্তুরাইজ করলে ভেজিটেবলস নষ্ট হয়ে যাবে। তুমি যদি 70 ডিগ্রি 72 ডিগ্রি নিয়ে যাও, ভেজিটেবলস গন, নিউট্রিশনাল ভ্যালু ডাউন হয়ে যাবে। সো অত হাই রাখা যাবে, বাট নর্মাল রেস্ট্রিক্টের থেকে আমরা একটু হাইয়ের দিকে রাখবো, হায়ার সাইড, দ্যাট মিনস 40, 42, 45। that considerable higher than the normal human body temperature, 5 to 6 degree higher. So temperature high रखले possible lab के select करा, only that one factor is possible enough to select the lab and eliminate the non-lab, because non-lab groups are more. Just lab is one group. See the listed of the microflora of my vegetables. Non-lab microbes are more inside the vegetables. So that one criteria is not enough. Needs other conditioning effect. So temperature one could be the factor. Another online students. What could be the criteria?
স্যার এখানে এরোবিক মাইক্রোফেল অ্যানারোবিক কন্ডিশনে রেখে দিচ্ছি।
Anaerobic রাখলে হ্যাঁ হবে possible, possible. This factor also could be correct. Anaerobic condition, lactic acid bacteria are facultative anaerobes. Is it?
হ্যাঁ
It is possible. It is possible because lactic acid bacteria facultative anaerobes. Facultative anaerobes. That means it can grow anaerobic condition also. But aerobic bacteria cannot grow without air, so they will be died or eliminated. Possible. Other factor, so দুটো ফ্যাক্টর পেয়েছি, টেম্পারেচার একটু হায়ার সাইডে রাখবো, এআর কে একটু কমিয়ে রাখবো, দ্যাট মিন্স অ্যানারোবিকের সাইডে রাখবো, লো এয়ার কন্ডিশন দেবো, অ্যানারোবিক কন্ডিশন রাখবো, দ্যাটস ওয়াই এরোবিক ব্যাকটেরিয়া কুড নট জেনারেট, কুড নট মাল্টিপ্লাই. Good point. Other factor that select lab only. Main point still missing. Main factor, that is salt, because lab prefer to grow in high salt, moderate salt, not high salt, moderate salt condition. But this non-lab bacteria eliminated at high salt concentration. That is why when pickles are made, so addition of salt দেবে, দেখবে একদম স্টার্টিং এই salt দিয়ে দেবে. Why? To eliminate or to degrow the other bacteria and uh, uh, increase the growth of lactic acid bacteria. So factoring that favor lab growth over non-lab, and that is important here because we are relying on the natural microbes. Factors that influence the growth of lactic acid bacteria over non-lactic acid bacteria. And this factor should keep in mind during the fermentation. So number one, temperature. Number two, salt concentration, very important. Number three, anaerobiosis. Anaerobiosis. So কোনটা কি elevated, কোনটা down রাখবো? Temperature elevated or down?
Elevated, but 40 to 49 normal body temperature.
Normal এর থেকে elevated. Elevated temperature. So try to ferment above 40 degrees centigrade, which will be very beneficial for lab to grow. Yes or no? And eliminate the growth of or slow down the growth of non-lab that temperature. But this one factor is not enough. Combined effect is enough to grow lab over non-lab. And second factor is very important, salt concentration. During the fermentation, the salt concentration should remain high, high salt. Because lab can accommodate the high salt concentration, but not the non-lactic acid bacteria. These microbes are potentially damaged at the high salt concentration. Damage or prevent the growth of non-lab, but high salt lab prefer to grow. So salt concentration is important factor. And third factor, anaerobiasis. Anaerobiasis means the oxygen concentration, that is the scenario here. Oxygen concentration should remain low or absent. In that condition, lab can grow because lab sustain to grow because lab is facultative anaerobes. In the absent or low, lactic acid bacteria can grow because it is facultative anaerobes, but other aerobic bacteria without oxygen cannot grow. Without oxygen, prevent the growth and metabolism. Without the air, first affected point is the metabolism. Metabolism will be slow down and will correspond to the growth also slow down. But lab can grow, can accommodate in low air condition also. So these three combined factors increases the growth of lab over the non-lab. And whenever the lab start to grow inside the vegetables, they produce acid. And this low acid, number 4, low pH. Another critical factor, when lab predominant over the other, labs start to grow and produce lactic acid, and this lactic acid make the overall condition pH down acidic side, and that acidic pH also inhibit the growth of other. So low pH another factor that influence the lab. So when lab predominate, they start to produce lactic acid and make the environment low pH acidic. And this acidic pH lethal for other bacteria. Point clear? How do you select lab over the non-lab in the vegetables-based fermented product? These factors should be keep in mind. So during fermentation, temperature should be elevated, salt concentration remains high, anaerobic condition influenced, and pH automatically down when labs are start to grow. Point clear? Next, though we are select lab over non-lab by conditioning the factors. Okay. So though we are select lab over non-lab by conditioning the environment, but in modern days, fermentation use starter culture, use starter culture over indigenous microbes.
बहुत efficient है.
So in modern day industry, fermented industry, starter cultures are directly used, not the indigenous microbes. But in previous era, they are using natural microbes by conditioning the environments like that, temperature, pH changes, etc. So starter culture for fermented vegetable products. What type of starter cultures are used now and what are their criteria? Starter culture for fermented vegetable product. Specifically, lab strains are used, specific, like pickle, leuconostoc mesenteroids, lactobacillus plantarum, lactobacillus brevis, pediococcus, pentoseus. These specific strains are used.
Sir, could you tell the spelling of the last strain?
Pentoseus, Soueus, Pentosaueus, Pediococcus pentoseus. আচ্ছা, যখন ন্যাচারাল বা ইন্ডিজেনাস মাইক্রোফ্লোরা লেখা ছিল তখন কোনো স্পিসিস দিয়েছি?
না।
অনলি জেনাস দিয়েছিলাম না? বিকজ ভেরিয়াস স্পিসিস ক্যান present in indigenous microbes. But when we choose starter culture, specific strain, specific species, we have to select. That's why তুমি চেক করো এখানে লিউকোনস্টা ওখানে লিখে ছেড়ে দিয়েছিল। But here we write here Leuconostoc mesenteroides, Lactobacillus specific species, Plantarum, which is important for vegetable-based fermented food, Lactobacillus brevis, Pediococcus pentosus, etc. Pickle fermentation is used. Next product, sauerkraut. In sauerkraut, we used Leuconostoc mesenteroids, same kind of same, Lactobacillus plantarum, Lactobacillus brevis, same, and Pediococcus pentosus. So why we select the specific species of that particular genus? Because this genus gives us suitable criteria for vegetable fermentation. That's why. So next topic should be specific genus and species. Genus to specific hobe, species also specific. Criteria to select starter cultures for vegetable based fermentation. First criteria, minimum nutrition requirement. Minimum nutritional requirement. You select two bacteria, bacteria A, genus, species A, Lactobacillus, species A, and Lactobacillus, species B. Lactobacillus A can easily grow in glucose, but Lactobacillus B need glucose during growth, as well as some need luxurious requirement, like it needs some good source of vitamins and other growth factor. Without that, Lactobacillus B cannot grow. Clear? So কোনটা সিলেক্ট করবে, A or B? না, ভেবে দেখো। মিনিমাম
এখানে
রিকোয়ারমেন্ট।
এ চুজ করবো।
Lactobacillus A can easily grow with the suitable glucose addition. Just add glucose and Lactobacillus A will grow easily. But B need glucose, need vitamins, need other factor to support the growth of bee. So bee কে grow করাতে গেলে আমার headache, as well as তুমি যদি মানির ইকোনমিক ব্যাপারটা ভাবো, A is economical because only you support the glucose support and he bacteria will grow easily. But B needs luxurious product, extra additional requirement. So economically not good for lactobacillus B to grow or to use. So that's why this point one should keep in mind minimum nutritional requirement bacteria we select during the fermentation. Point clear? This point is clear? Minimum nutritional requirement. Everyone. Number two, tolerate acids. Another criteria. Because during the growth, fermented products are acids, various acids including lactic acids and lactic acids create the environment's pH lower side. So that lower side where the bacteria should remain alive. Because bacteria যদি death হয়ে যায় low pH এ, তো fermentation করবে কি? Point clear? Point number two, tolerate to acid. So the bacteria genus that species that we select here, it should be acid tolerant. Yes or no? Answer. Acid tolerant. Bacteria should be acid tolerant. Yes or no?
Yes.
Audio clear তো?
হ্যাঁ স্যার।
হ্যাঁ, answer করুন। Number three. Able to ferment diverse carbohydrate.
স্যার ভিডিও খুব হেজি আসছে, সঠিকভাবে দেখতে পাচ্ছি না।
আবার ভিডিও এসি।
হ্যাঁ স্যার, অনেকক্ষণ দিয়েই দেখা যাচ্ছে না, প্রথমে যখন এগুলো লিখছিলেন তখন থেকে।
কি করা যায়?
স্যার আপনি একটু পিডিএফ করে পাঠিয়ে দেবেন, কি লিখছেন ভালো করে দেখা যাচ্ছে না।
আমার তো পিডিএফ এই মুহূর্তে পসিবল নেই। ক্লাস নোটসে আমি কি করে পিডিএফ করব? কেউ যদি স্ক্রিনশট নিয়ে থাকে সেখান থেকে পিডিএফ পসিবল। আর আমি তো নোটস এমনি দেবো, সেখান থেকে পেয়ে যাবে, বাট জিনিসগুলো বোঝার জন্য আর কি। যদি বুঝতে চাও বুঝে নাও, লেখাটা ছেড়ে দাও, লেখা নোটসে পেয়ে যাবে। যতটা লিখতে পারছো লেখো, আদারওয়াইজ নোটসে পেয়ে যাবে। এখান থেকে পিডিএফ আমি করতে পারবো না। তো যেগুলো
লাস্টের পয়েন্টটা কি লিখেছেন?
able to ferment diverse carbohydrate. দেখো, vegetables contain variety of carbohydrate including monosaccharide, polysaccharides, and majority time the carbohydrate found in the vegetables are polysaccharide, that means complex sugar, complex carbohydrate. Many of this time the vegetables contain complex carbohydrate, that means polysaccharides. So তুমি microbes select করলে, that microbes is not enough to digest polysaccharides. তো microbesটা কি কোনো কাজ হবে, fermentation দেবে?
No.
That's why আমি criteria লিখে রেখেছিলাম। Able to ferment diverse carbohydrate, that means the microbes that I select, it should use monosaccharides, and also they can use the polysaccharide also, that means complex sugar they can also use, and that microbes we are selecting because vegetables contain many of times complex carbohydrates, and during the growth need to break down this complex carbohydrate into simple one. Third point clear? Number 4 criteria: non-proteolytic. Non-proteolytic. The species that I select during the fermentation of vegetables should be non-proteolytic, because if the bacteria is proteolytic, that means they releases proteases and degrade the protein material. And if protein material is degraded inside the vegetables become spoiled. Then vegetables are spoiled. তোমরা খেয়াল করবে বাড়িতে কিছুদিন যদি ভেজিটেবলস রেখে দাও কি হয়ে যায়, সফট রটেন বলে, এটা একটা ডিজিজ, এটা একটা স্পয়েলেজ টাইপ। সফট রট মানে সফট হয়ে যায় না জিনিসটা, আর খাওয়া যায় না। যে কোনো ভেজিটেবলস যদি 2-3 দিন তুমি উইদাউট ফ্রিজ রেখে দাও কি হবে, সফট হয়ে যাবে। হ্যাঁ, সো দ্যাটস কল সফট রটেন প্রবলেম, স্পয়েল, মেনলি ডান বাই প্রোটিওলাইটিক ব্যাকটেরিয়া। প্রোটিওলাইটিক ব্যাকটেরিয়া গ্রো করে আর ওটাকে সফট করে দেয়। এবার তুমি ব্যাকটেরিয়া স্পিসিস নিচ্ছ যে প্রোটিওলাইটিক, তো তো স্পয়েল করে দেবে, তো ফার্মেন্টেশন তো দূরের কথা, স্পয়েল করাবে গিয়ে। সো অবভিয়াসলি the microbes should be non-proteolytic. Number 4. Number 5. Produce desired flavor. Produce desired flavor. Yes, that is also correct. Bacteria should not produce off flavor compound. Off flavor compound. Should not produce. Number 6, tolerate to salt. The bacteria that we are selecting should be tolerable of salt concentration, high salt concentration. Number 7, able to compete with other organisms. As vegetables contain indigenous microbes also, that's why when we add the starter culture, this starter culture have competitiveness. That means it grow faster, That's why the other bacteria are lag behind and start to stop the growth or eliminate it. So this competitiveness must remain inside our starter culture organism. High growth rate. Then other microbes. Then it is possible only to starter culture ওখানে গ্রো করবে, বিকজ হাই গ্রোথ রেট মানে কি কুইকলি গ্রো করছে ওভার দি আদার মাইক্রোবস। দ্যাটস ওয়াই পুরো মাইক্রোবস এর যে ইয়েটা হবে, পুরো যে ইকোলজিটা হবে সেটা পুরোই স্টার্টার কালচারে হবে, এন্ড ইন্ডিজিনাস মাইক্রোবস আর থাকবে না। So that's called competitiveness, able to compete with other organisms. So these 7 criteria should remain keep in mind during the selection of starter culture in vegetable based fermented product. Clear? নেটওয়ার্ক ইস্যু হচ্ছে, আজকে আর এগোচ্ছি না। নেক্সট ডে only 2 products are here, vegetable based fermented product, pickle and sauerkraut. নেক্সট ডে কমপ্লিট করব ইউনিট। সো আজকে এত অবধি রাখবো, এনি কোশ্চেন থাকলে করতে পারো।
টলারেট টু অ্যাসিডটা বোঝাবেন?
টলারেট টু অ্যাসিড না টলারেট টু সল্ট?
হ্যাঁ স্যার, টলারেট টু অ্যাসিডটা।
Tolerate to acid. So when microbes are grow, then pH will be down because lactic acid and other acids are produced during fermentation. Yes or no?
Yes.
So
Yes, sir.
pH down এ যদি আমি যে সিলেক্টেড স্ট্রেনটা নিলাম, pH down এ যদি ও নিজে কিল্ড হয়ে যায়, তাহলে ফার্মেন্টেশনটা অনগোয়িং করবে কে? আমার তো ফার্মেন্টেশন করতে হবে 48 আওয়ার, তোমার 36 to 48 আওয়ার, মানে 2 days 3 days। ভেজিটেবল ফার্মেন্টেশন নিডস টাইম, ঠিক আছে, 2 to 3 days আরামসে লাগে। সো যদি স্টার্টিং এ কিল্ড হয়ে যায়, তো 2 থেকে 3 days টানবে কে ফার্মেন্টেশন? সো শুড অ্যাবল টু টলারেট দ্য লো অ্যাসিড এনভায়রনমেন্ট, sorry, high low acid na high acid environment. That's why the selected strain should be tolerant to acid and also tolerant to salt, because during the fermentation আমরা salt add করি good concentration, so salt toleration ও দরকার।
Sir, in the last point, able to compete with other organism. Sir,
Correct.
the other organism are the organism which have to be eliminated or the organism which produce lactic acid?
No, these other organisms are the non-lab, which are the indigenous microbes, so non-lab should be eliminated and establish only our starter culture organism. That is why the growth rate of the starter culture organism should be high enough to establish the only starter culture organism and eliminate the others. And this competitiveness is crucial. Clear my point?